And we ended last time talking about an action potential, and just a quick review here, and hopefully the PowerPoint you can see in front of me is what you guys are seeing as well. We're going to kind of begin with the threshold and the all or none response. So what this has to do with is we talked about how the neuron needs to become depolarized to produce an action potential. And that means that the neuron has fired or a nerve stimulus has gone through it. But not all depolarization events will produce an action potential if the stimulus is not strong enough. For the axon to fire an action potential, depolarization must reach a threshold voltage to trigger that action potential. At threshold, the membrane is depolarized by 15 to 20 millivolts. So this means at resting, we were at about negative 70 millivolts. So that's what the inside of the cell is usually always at. And we need to depolarize the inside of the membrane at least to negative 50 or negative 55, which is a change about 15 to 20. When that happens, we call that the threshold point. And when that happens, the sodium permeability increases. So this is like the sodium starts to come in, and if the inside of the cell reaches threshold, then all of the sodium channels open and all of the sodium rushes into the cell, bringing with it its positive charge and making the inside of the cell more negative. So at this point, the sodium influx will exceed the potassium exiting, efflux. And this positive feedback cycle begins at threshold, meaning once we reach this threshold voltage, the action potential will fire no matter what. So we call that the all or none phenomenon. Once we reach threshold, an action potential will happen completely. The propagation of an action potential. Propagation allows the action potential to be transmitted from the origin, so where the depolarization began from a stimulus in the neuron, down the entire axon length towards the terminals or the end branches of that axon. The sodium influx through its voltage gates in one membrane area will cause local currents that cause openings of other sodium voltage gates in adjacent membrane areas that are downstream from it, you could say. This will lead to a depolarization of that area, which in turn will cause depolarization in the next area. So this, once initiated, an action potential will be self-propagating, meaning it will continue to pass down the axon by depolarizing every sodium channel that it passes. In non-myelinated axon, each successive segment of membrane depolarizes, then repolarizes, and the propagation in myelinated axons will differ a little differently. If you remember, myelination helps this signal to travel faster because of the myelin sheaths. Since the sodium channels closer to the action potential origin are still inactivated, no new action potential is generated there. So right at the origin of the stimulus in that neuron, another action potential cannot be generated exactly at the same time. So that helps for the action potential to continue down the same way. So it continues one way down the neuron. And it also is helpful so that the action potential doesn't go back the other way. And that's due to these sodium channels becoming inactivated for a few milliseconds right after depolarization occurs. So the action potential only occurs in a forward motion or forward direction. So here's the voltage at 0 milliseconds. So right before a neuron has been stimulated, we're at resting membrane potential, negative 70. We have a little recording electrode, which is recording the inside voltage right inside the membrane. And we'll see here that this is the direction of the action potential. The peak of the action potential is shown in red, and the yellow is the resting. So it's just showing how this is where the action potential will begin, and it's only going to flow down one direction. At about 2 milliseconds, we get our kind of peak, our action potential peak. And you'll notice here that behind the red mark is a hyperpolarization state. And that's because after the action potential has passed this area, this part of the neuron goes extra negative into that hyperpolarized state. But right here at about 2 milliseconds, we're recording the action potential peak right here at this electrode. Again, downstream, that part of the neuron is still resting. And here we are showing the repolarization state. And again, this gray area shows the portion of the neuron that's in the hyperpolarized state. And while that portion of the neuron is in the hyperpolarized state, again, that means it's extra negative after the repolarization period, another action potential cannot be generated because the sodium channels are inactive. So these are kind of the propagation of an action potential and how it travels down the axon in one direction. So this all occurs in 4 milliseconds, which is extremely fast. So it just goes to show how quickly your nervous system responds. This shows the propagation of an action potential, an animation that you can watch on your own. Coding for stimulus intensity. All action potentials are alike and are independent of stimulus intensity. The central nervous system tells the difference between a weak stimulus and a strong one by the frequency of impulses. And the frequency is the number of impulses received per second. So a higher frequency, so higher numbers of action potentials will mean there was a stronger stimulus being applied. So here is just a look how a sub-threshold stimulus will not generate an action potential. So here's the neuron at resting, and this is showing the voltage of the stimulus being applied. And again, the stimulus is any sort of stimulus that your body would perceive, whether it's a touch, a pain receptor, your photoreceptors in your eyes. If the stimulus that your body's perceiving, whether externally or internally, is not strong enough and it won't reach that threshold, then we won't generate an action potential and your body won't receive any sort of recognition of that stimulus. But here we have the stimulus reaching that threshold voltage, so we'll fire off action potentials. The stronger the stimulus means there will be more action potentials generated. But you'll notice that the action potentials, the inside of the cell, never gets past about positive 30. But if we have a stronger stimulus, that just means we're generating more action potentials. The refractory period then. So the refractory period refers to the time in which the neuron cannot trigger another action potential. And refractory periods are important because there's always a part of the neuron that's inactive in its refractory period. And that's so that the action potential only travels down the way it comes from and it doesn't jump back the opposite way. During the refractory period, the voltage-gated sodium channels are open, so the neuron cannot respond to another stimulus. They'll be inactive, so that when it says they're open here, they're in an inactive state. There's 2 types of refractory period. The absolute refractory period is the time from the opening of the sodium channels until they reset back into their active state. This ensures that each action potential is an all or none event, meaning when it reaches threshold, it'll generate an action potential or it won't at all. And this also enforces one-way transmission of nerve impulses. The relative refractory period follows the absolute refractory period. This is where most sodium channels have returned to their resting state, but some potassium channels might still be open and repolarization is still occurring. The threshold for the action potential generation would be elevated due to the fact that we're most likely in our hyperpolarization state. The only way to generate an action potential during this period is if we have an exceptionally strong stimulus. And they give you an example of a disobedient refractory dog. Think of a disobedient dog. If he is absolutely refractory, he will never come when called. But if he is relatively refractory, he may come, but only if you call loud enough. So again, 2 types of refractory period. The absolute refractory period, this is the time from the opening of sodium channels until the resetting. So no action potential could be generated during this refractory period. The relative refractory period follows that. where some sodium channels have returned to their state. So we could generate an action potential if the stimulus is strong enough. So here's a look at where the absolute refractory periods will be and where the relative refractory periods will be. Again, the absolute refractory periods will be when the sodium channels, they will depolarize the cell, but then they'll go into their inactive state. So then this area of the neuron cannot be stimulated again. Whereas the relative refractory period is when the neuron is in the hyperpolarized state, meaning there's sodium channels that could be stimulated again, but the neuron is really extra negative. It's about -90. So we need that much more jump in voltage to reach a threshold to get an action potential peak. Any questions on that? Okay, conduction velocity. This will have to do with the axon, the diameter of the axon, and the degree of myelination. Action potentials only occur in axons, not in other cell areas, so their conduction velocities will vary widely, depending on how large the diameter is of the axon and how myelinated it is. So a larger diameter fiber has less resistance to any local current flow, so a large diameter axon will have a faster impulse conduction. And there's two types of conduction depending on the presence or absence of myelin. And we call one of them, these two types of degrees of myelination, continuous conduction or saltatory conduction. Saltatory has to do with jumping. I think in Spanish, saltar is to jump, if I remember. So there's some Greek, Latin roots there, but saltatory means jumping. So continuous conduction is slow conduction that occurs in non-myelinated axons. So this will be extremely slow. The saltatory conduction is about 30 times faster. This occurs in axons that are myelinated. The myelin sheaths will insulate, prevent leakage of the charge, and the voltage-gated sodium channels are located in the gaps between myelin sheaths. So the action potential will only be generated where those sodium channels are. So the electrical signal will appear to jump rapidly from gap to gap. So this is showing here the stimulus being applied, the size of the voltage. This is in a non-myelinated, I believe. In non-myelinated axons, the conduction will be very slow. This is called continuous conduction. Whereas in myelinated axons, the conduction is fast. The myelin keeps the current in the axons, so there's not much voltage decay, we say. So the action potentials are generated in the gaps, and they will jump wherever there's sodium channels, which are these things here. Some clinical relationships with what we've been talking about: multiple sclerosis. It's an autoimmune disease, affects primarily young adults, mainly women, not always. Multiple sclerosis is when the myelin sheaths in the central nervous system, so the brain and spinal cord will be destroyed when the immune system attacks itself, its own myelin. It turns the myelin into hardened lesions called scleroses, and the impulse conduction will be incredibly slow due to these hardened lesions. So demyelinated axons increase sodium channels, causing cycles of relapse and remission. No cure for MS, but there are some things you can take to manage it. Symptoms: visual disturbances, weakness, loss of muscle control, speech disturbances, incontinence. So wherever you have a muscle, and kind of a loss of just conduction speed within that muscle. Treatment: drugs that could modify your immune system activity may not be able to totally prevent, but maintaining high blood levels of vitamin D may reduce risk of development. So another reason to take vitamin D. Conduction velocity, they fall into three groups. So these nerve fibers are classified according to how large their diameter is, their degree of myelination, and the speed of conduction. We say group A fibers are the largest diameter. They're myelinated somatic sensory and motor fibers of skin, skeletal muscles, and joints. And this is the speed of conduction. And I think this is interesting. It passes on their electrical signals at about 300 miles per hour. Isn't that crazy to think about, that that's going on in your body all the time as your photoreceptors that you're seeing in right now are doing this. Group B fibers are the intermediate. They have about a conduction speed of 30 miles per hour. Group C is the smallest diameter, and they will be completely unmyelinated. B and C will include some autonomic nervous system visceral motor and sensory fibers that serve visceral organs. A little more clinical aspects: impaired action potential impulse propagation can be caused by a number of physical and chemical factors. Anesthetics will act by blocking some of those voltage-gated sodium channels, so to try to keep pain from traveling so you feel it. Cold temperatures or continuous pressure will interrupt blood circulation and delivery of oxygen to the neurons. So your cold fingers will get numb, or your foot will feel like it's going to sleep. And if you notice, if you ever feel those kind of like prickling on your foot, that's just the interruption to blood circulation and delivery of oxygen to the neurons. So you'll feel a different sensation. Okay, so what we're going to do is just kind of finish this chapter, and the part C goes into a lot of detail, and I'm just going to show you what I want you guys to know from part C for this chapter, and then we'll be done. And because I know each chapter has like 500 PowerPoint slides, I feel like. We're going to briefly talk about the synapse in the nervous system, because at the synapse, we have different drugs that will attack receptors in the synapse. So we won't learn all the details, but it's important to just understand why the synapse is a really integral part of your nervous system. And the synapse is the space between neurons. So where one neuron is connected to another neuron, or where one neuron is connected to an effector cell, whether that's a muscle, a gland, an organ. So the nervous system works as information flows from neuron to neuron, and neurons will be functionally connected by synapses, which are junctions that are unique to the nervous system that mediate this information transfer. They're from one neuron to another, or from one neuron to an effector cell. The presynaptic neuron is the neuron conducting impulses towards the synapse. So we call this is the neuron that sends information. And the postsynaptic neuron, post, after the synapse, is the neuron transmitting the electrical signal away from the synapse. So this neuron will receive the initial information and then send it down. In the peripheral nervous system, the postsynaptic neuron could be another neuron, or it could be a muscle cell or a gland cell. And most function as both presynaptic and postsynaptic, because there will be many neurons in a line. So we have different types of synapses. Axosomatic will be from an axon to a soma, a cell body. Axodendritic will be a connection between an axon and a dendrite. And an axoaxonal will be a connection from one axon to another axon. So here are synapses underneath some sort of microscope at incredibly high magnification, just showing the synapse, or the connection of an axon. This specifically is an axosomatic synapse because it's connected to the cell body of another neuron, a postsynaptic neuron. So kind of a cool picture there. So these are the synaptic connections that I just described: the axodendritic, axosomatic. These are less common ones: an axon to axon connection, or dendrite to dendrite. There's two main types of synapses. They'll be chemical synapses or electrical synapses. The chemical synapses are the most common type. These are specialized for release and the reception of chemical neurotransmitters. And you have many neurotransmitters in your body. They're typically composed of two parts: the axon terminal of the presynaptic neuron. The axon terminal, this is where the axon will branch and end. It will contain synaptic vesicles, and these little tiny vesicles or bags are filled with a neurotransmitter. Then the receptor region on the postsynaptic neuron's membrane will receive that neurotransmitter once it's been released. And usually it's on a dendrite or a cell body. And these two parts, the pre and postsynaptic neuron, are separated by a fluid-filled synaptic cleft. The electrical impulse will be changed to chemical across the synapse and then back into electrical. So it's kind of interesting to see how this all works. So how do we get transmission of a neurotransmitter across this synaptic cleft or space? The synaptic cleft prevents the nerve impulses from directly passing from one neuron to another. There's a chemical event. So instead of just passing along an electrical signal neuron to neuron, we have to bring chemicals into this. And the transmission will depend on the release, the diffusion, and the receptor binding of the neurotransmitter. And this will ensure unidirectional communication between neurons. So everything is extremely controlled. There's different drugs that will affect receptor binding of certain neurotransmitters, how well they're diffused, or how well they're degraded. So a lot of drugs that we take specifically affect receptors on these postsynaptic neurons, because any sort of drug that inhibits the chemical from being released or received, or from the electrical signal from continuing to being passed, will completely stop whatever action the nervous system was trying to have. So this animation will show how the vesicles are released from the presynaptic neurons, how the neurotransmitter will be released from the vesicle and will be received by receptors on the receiving cell or the postsynaptic neuron. So this information transfer, there's 6 steps that are involved, and this is kind of all we're going to be covering in this PowerPoint. We'll be covering these 6 steps, and we won't go into too much detail outside of these 6 steps. So for your studying purposes. The 6 steps are the action potential. So after we go through all that depolarization, repolarization, hyperpolarization that you guys are probably sick of hearing about, the action potential eventually comes to the end of that axon. So it comes to the end of the line, and that is the presynaptic neuron. When the action potential arrives at the end of the axon terminal, voltage-gated calcium channels open and calcium enters the axon terminal. Calcium flows down its electrochemical gradient from the extracellular fluid, so outside the neuron, to the inside of that axon terminal. So here's the first step. The chemical synapses transmit signals from one neuron to another using neurotransmitters. And it's hard to read here, but this is showing, let's see if we get any, we don't get any words, and these are hard to see, but it's saying how the voltage-gated calcium channels are opening and calcium enters in this axon terminal. Step number 3, calcium entry causes synaptic vesicles to release the neurotransmitters. So simply put, when the calcium comes into the axon, it causes those vesicles to release the neurotransmitter. How that happens is pretty much the vesicle will fuse, and it results in an exocytosis-like transport of the neurotransmitter into that synaptic cleft. The higher the impulse frequency, the more vesicles will be released, leading to a greater effect on the postsynaptic cell. So a greater frequency of action potentials will release more acetylcholine or release more neurotransmitter to try to get the signal across to the next cell faster. So now the calcium causes these vesicles to fuse with the membrane and exocytose the neurotransmitter which is in them. The neurotransmitter will then bind to receptor proteins on the postsynaptic neuron. So the neurotransmitter diffuses, it crosses over the synaptic cleft, and it binds to a specific receptor that's especially made just for that neurotransmitter. It's often a chemically gated ion channel, meaning that once it binds, a different ion will enter the postsynaptic cell. Binding of the neurotransmitter opens the ion channel, creating another graded potential. And you remember, a graded potential is like an action potential, but it only happens in usually one spot and it doesn't travel as far. The binding causes the receptor protein to change shape, which causes the ion channels to open, and this causes a graded potential in the postsynaptic cell. This can excite or inhibit the cell. So this can be an excitatory response or some sort of inhibitory response as well. So this just shows the binding of the neurotransmitter to these receptor proteins. And I'm so sorry, these pictures are so small, but it's showing once that receptor binds, once the neurotransmitter binds, it'll usually cause a different ion, normally sodium, to start the depolarization process all over again on the next neuron. The neurotransmitter effects are eventually terminated. So we don't want the neurotransmitter to completely or to constantly being received at the postsynaptic cell. So as long as the neurotransmitter is binding to the receptor, graded potentials will continue to fire. So that means that the postsynaptic neuron will continue to be stimulated because sodium will be rushing into it. Within a few milliseconds, the neurotransmitter will be terminated by reuptake by astrocytes, degradation by specific enzymes, or diffusion away from the synaptic cleft. So this process is very well controlled. How scientists figured this all out in a lab, years ago, it's just amazing how far science has come and what great things we can do now in terms of human health because of it. So this, again, these pictures are really small, but it's showing the different ways that the neurotransmitter gets either taken back up, degraded, basically so it's not constantly causing a graded potential on the postsynaptic neuron. There's synaptic delay, which is the time needed for a neurotransmitter to be released. The synaptic delay is what we call the rate limiting step of neural transmission, because synaptic delay, the rate limiting step, what that just means is how quickly the acetylcholine goes across will determine how fast this transmission can occur of the electrical signal. Okay, and then these postsynaptic potentials, again, all you need to know is that the neurotransmitter and the signal that it can create, it can be excitatory or it can be inhibitory. And I'm going to skip over, this takes you through a lot of, if you are interested in learning that, you can work your way through those slides, but we're going to pass over that because I think chapter 11 has enough for you to focus on. But if you are interested in how different, like carbon monoxide, nitric oxide, some gases, some drugs have inhibitory effects, what they're doing is they're having effects on these receptors on the postsynaptic. So it's interesting stuff, but I think we have enough to worry about in this chapter. We're going to close this chapter, and it's only 7 o'clock, so maybe we'll start chapter 12. We'll close this chapter just briefly talking about reflexes. So patterns of neural processing. And this just is talking how your neurons travel throughout your body. Serial processing is that when the input travels along one pathway to a specific destination. So usually one neuron stimulates the next one, which stimulates the next one. The system will work in an all or none manner to produce specific anticipated responses. And a best example of a serial processing is a spinal reflex. And a reflex, these are very interesting. They are involved in what we call a reflex arc. A reflex is a rapid automatic response to some sort of stimuli. There are particular stimulus that always causes the same response. So you have different pathways in your body that create an automatic response. So it's extremely quick. And we call these different pathways reflex arcs. There will be 5 components to a reflex arc: the receptor, the sensory neuron, the central nervous system integration center, which sometimes is just your spinal cord, the motor neuron, and then the effector. And this is a look at a simple reflex arc, and we might go through this more in detail later on in the nervous system, but I wanted to just briefly introduce it now. So the 5 steps of a simple reflex arc, or a stimulus will cause a receptor to send a signal down a sensory neuron to the integration center. And a lot of times, simple reflex arcs will just be in the integration center, will occur in the spinal cord, and it will send out an immediate automatic response through the motor neuron to the effector. And a great example of this is you have several different types of reflexes, but probably the easiest one is just a simple pain reflex. If you touch a hot stove, you immediately withdraw your arm, and that's because it's the simple reflex arc. A stimulus like heat or pain will cause that receptor to send a signal to your spinal cord. Sometimes it goes up to your brain. It'll send out a motor response immediately to the muscles in your forearm to take your hand off the hot stove. So that's a simple reflex arc. Okay, questions about that at all? All right, so I'm just gonna briefly introduce chapter 12, especially maybe just part A of chapter 12, because a lot of it is just anatomy of the brain. And give me 1 second to pull that up here. And we'll just continue from there. Any questions on lab? Okay. All right, thanks for being with me. Just pulling up. No questions. Thanks, guys. All right. Okay, so we're just gonna start the central nervous system. I know it's been pretty heavily focused the last couple lectures, but we're just gonna start a little review in the brain just to see how much you know, because this will be a good introduction to the central nervous system. So your central nervous system consists of your brain and spinal cord. Cephalization is the evolutionary development of the anterior portion of the central nervous system, resulting in increased number of neurons. It's the highest level reached in the human brain. This is going to go through brain development from the embryology, and I'm going to skip over embryology because that could be its own separate class. In grad school, I took a embryology course. So anything related to embryology, you guys can skip over or brain development. But let's get to the adult brain. The adult brain has 4 regions: the cerebral hemispheres, the diencephalon, and then the brain stem, which consists of the midbrain, the pons, the medulla oblongata, and the cerebellum. So these are the different parts: the cerebral hemispheres, the diencephalon, we'll talk about what makes that up. The cerebellum is a separate part of the brain in the back. It sits in the posterior fossa of the floor of your skull. And here's your brain stem, starting with the midbrain, the pons, and the medulla oblongata. The gray matter are short non-myelinated neurons. So gray matter is non-myelinated and white matter is myelinated. Could include some non-myelinated axons as well. The basic pattern found in your central nervous system is you have a central cavity, which is usually filled with cerebral spinal fluid, surrounded by gray matter with white matter external to the gray matter, and it makes this butterfly type structure when it's in the spinal cord itself. Okay, so if we take a look just at a cross section and the distribution of gray and white matter, this is showing a cross section of the spinal cord. You'll notice here the central cavity travels up and down. That will contain cerebral spinal fluid. The inner gray matter makes this butterfly-like structure, and the outer white matter surrounds it. And again, the gray matter is made up of neuron cell bodies, and the white matter is mostly myelinated axons. The brain stem has additional gray matter nuclei that are scattered throughout it, and the cerebral hemispheres, also known as the cerebrum and the cerebellum, contain an outer layer of gray matter called the cortex. So if we take a look at a cross-section of the cerebrum, and we take a look at it here, you can see the central cavity shown in this picture is the 4th ventricle. You can see here the inner gray matter and the outer white matter, and just how the gray and white matter is distributed throughout the brain. Another look at the brain, this is a look through, I guess, a frontal coronal section. You can see the gray matter forms the cortex on the outer layer of the brain with the white matter more inside. Okay, ventricles, then you have ventricles throughout the brain. These are fluid-filled chambers. They're continuous one another and to the central canal of the spinal cord. So because they're continuous with one another, they're all connected, they will all contain CSF, the cerebral spinal fluid. They're lined by the neuroglial ependymal cells. You have two C-shaped chambers that are the lateral ventricles, and that pair is separated by the septum pellucidum. You have a third ventricle, and then you also have a fourth ventricle, which is connected to the third by the cerebral aqueduct. So hopefully this picture is kind of familiar to anatomy, that you remember a little bit where these ventricles are located. And again, these ventricles are filled with cerebral spinal fluid. They provide buoyancy to the brain and also allow for communication of the cerebral spinal fluid throughout the whole spinal cord, because it's all connected completely down the spinal cord in the central canal. The cerebral hemispheres form the superior part of the brain, and these are what you probably think of when you think about your brain. They account for 83% of your brain mass. You have different surface markings. Gyri are the ridges, the sulci are the grooves. There's fissures. The longitudinal fissure separates the right from the left hemisphere, and the transverse cerebral fissure separates the cerebrum and the cerebellum. So here's the longitudinal fissure. You can see here cerebral veins and arteries. You can see the gyri are the brain folds, and any sort of sulcus is a depression or a valley between those folds. Here is a look at the lobes of the brain: the frontal lobe, parietal lobe, occipital lobe, temporal lobe. The brain stem will be directly anterior to the cerebellum, and again, the transverse fissure separates the cerebellum from the cerebrum above it. And again, you probably have some sort of familiarity that different things occur in each lobe. So that's why I want us to have a good understanding of what the lobes are. Several sulci, or deep depressions, divide each hemisphere into the 5 lobes. The insula is a lobe that's kind of in the within. You have to pull back the temporal lobe to see the insula. The first 4 lobes are named after their overlying skull bones, and the insular lobe is buried underneath portions of those 3. So this is a great animation to review the anatomy of the brain if you want to go over that on your own. Here is a look at the different lobes. And again, the sulcus that will separate the lobes from each other. This just shows you kind of a cutout that shows you the fissure, which is a deep sulcus separating out the blue lobe from the pink lobe, or the red lobe, which is your temporal and frontal lobe. So again, a lot of this is review, but hopefully this just gets your, it whets your appetite for more exciting things to come in the brain central nervous system PowerPoint. But there's your insula within kind of the brain underneath those lobes. I'm gonna pass through a little bit of this now, because this is a little more cerebral cortex. This is the executive suite of the brain. It's the site of conscious mind. So all of where your awareness, sensory perception, voluntary motor initiation, communication, memory storage, and understanding occurs. This will be the thin, most superficial layer of gray matter. This makes up about 40% of the brain mass. Functional imaging like PET and MRI scans of the brain shows that a specific motor and sensory functions are located in discrete cortical areas called different domains, and higher functions will be spread over many areas. So this shows a functional neuroimaging of the cerebral cortex, and again, just the most superficial layer of the brain. And it's showing just where your sense of seeing, hearing, speaking, and thinking, and where those functions will occur throughout the brain. So it's amazing that with a functional MRI, we can see when that part of the brain is being stimulated on this sort of imaging. The 4 general considerations of the cerebral cortex, they contain 3 types of functional areas: the motor area, the sensory area, and any sort of association area. The motor area will control voluntary movement, and the sensory area will control all sort of sensations that will be sent up to the brain. So the conscious awareness of sensation. Each hemisphere is concerned with the contralateral opposite side of the body. That means the left side of your brain senses and controls things on the right side of the body, and vice versa. Lateralization is specialization of cortical function can occur in only one hemisphere, and conscious behavior involves the entire cortex in one way or the other. And we'll be done soon, but I want to get through a little bit of the motor and the sensory areas, which again, I hope you touched on in anatomy. The motor areas are located in the frontal lobe. They act to control voluntary movement. And the primary motor cortex is located in our precentral gyrus. The premotor cortex will be anterior to that precentral gyrus. So I just want to talk a little bit about here. These are your motor areas in the frontal lobe, and the primary motor cortex is located in this precentral gyrus. And the precentral gyrus refers to the brain fold that's directly anterior to the central sulcus. And this is your central sulcus or main kind of indentation in your brain folds. So I want you to know where that primary motor cortex is located in that precentral gyrus. This tells you also where your frontal eye field is, the Broca's area, the anterior association area, but the big one again I want you to know is the primary motor cortex. And then we compare that with the sensory areas. The primary somatosensory cortex is located in the postcentral gyrus, just directly behind the central sulcus. So those two, I like you guys to know, the primary motor cortex, where that's located, and the primary somatosensory cortex, where that's located. You'll also see more association areas here, where vision is, where hearing is. The visual association area will be located in the back, in the occipital lobe. The auditory association area is located near the temporal lobe. You have taste and equilibrium. So taste is gustatory. Equilibrium has to do with balance, especially with the fluid in your ear. That's located in the insula lobe. And again, that was the inner lobe of the brain. So this takes us through the primary somatic motor cortex, located in the precentral gyrus, again, of the frontal lobe. Somatotopy are where all muscles of the body can be mapped to an area on the primary motor cortex. And a motor homunculi is an upside down caricature drawing that represent these contralateral motor innervation of body regions. And I just want to talk through this a little bit because this is really interesting to see. So this is just a caricature drawing that is accepted widely by scientists, medical professionals, and it explains the part in the cortex, so the part in that outer layer of the brain in the gray matter, and where it will control what part of the body. And again, it's always contralateral. So this is the left motor map in your precentral gyrus, and you can see how much of the cortex is given for different parts of control in the body. So you'll notice a large part of your brain in this cortex controls your fingers and hands. And you have a lot of sensory receptors in your fingers and hands and a lot of fine motor control there. So here's your motor control, a lot of motor control in your face as well, swallowing, tongue. You'll notice your knee and foot and toes get hardly any motor control area, but there's a lot of attention given to your hand, fingers, and face. So again, this side, the left side of the brain, will control the actions on the right side of your body. Same thing, the sensory map in the postcentral gyrus. This will control the sensations received, so where all of your sensory input is coming from. Again, that's coming from the left side of the body will be sent up to the right side of the sensory map in the postcentral gyrus. And again, you notice that your hand and fingers get a huge part of your brain kind of sensory map in relation to your knee, your leg, your hip. There's just a tiny part of the brain for those. And again, that's because there's a lot of sensory nerve endings in your fingers. So I think that's kind of cool. So I'm going to have you guys learn that too. I think it's applicable. More about the cerebral cortex. I think what we'll do is we'll probably end there with the cerebral cortex. And again, just reviewing The big thing about the cerebral cortex is I want you guys to know the precentral and postcentral gyrus contain the primary motor cortex and the primary somatosensory cortex, and it can be depicted by this caricature drawing called a homunculi. See, even I can't say these words. So we will pause there